Now, for the second time, Professor Roger Barker is going to speak to us about cell and gene therapy. Welcome, Roger.
Well, thank you very much for inviting me and to talk twice. This is a very rare event. I feel somewhat inadequate after hearing all of those inspirational stories. So what I'm going to do in this talk is take you through the current status of dopamine cell therapies and gene therapies for Parkinson's. And this is a topic that goes in and out of fashion. So this has been going on for probably 40 years. And I always say to my junior research people, pick a topic and stick with it. And at some point in your life, it will be flavour of the month. And at other points, people will be amazed you're still working on it. So this is a topic that has come in and out of fashion. It's also fair to say, especially around cell therapies, that there is an awful lot of nonsense out there with cell therapies. So you have to be a little bit careful about some of the things that you read about. So I'm going to start probably with nothing. Well, I'm going to start with gene therapies, if the slide ever changes. I can't seem to get it to move despite pressing the button. And I'm going to talk about the different types of gene therapy that exist. So essentially, the way to think about gene therapies for Parkinson's is around a number of key concepts. So at the heart of Parkinson's, as we've heard, is the loss of this dopaminergic population of cells in the substantia nigra, which project up into a part of the brain called the striatum. And importantly, for a lot of what I'm going to discuss, that is quite a small population. So you have about 500,000 dopamine cells on each side of your brain, and when you lose half of those, 250,000, you get the first motor features of Parkinson's, and they probably also contribute to some of the other problems around thinking. So 250,000 dopamine cells, if you can get that, you're probably going to be all right. So that's the ambition, and that is tractable. So how could we conceive about doing that with gene therapies? Well, one way is to try and say, let's regrow that system. So let's accept that the dopamine system is dying off. Can we give it some sort of fertilizer which will encourage it to grow? So we'll put something in and regrow that pathway in people with Parkinson's. So that's one approach. A second approach is to say, I'm not going to regrow it. It's going to die. It's going to go. What I'm going to do instead is I'm going to inject a gene into your brain, which is going to go into the cells where the dopamine normally works. So in this place called the striatum, it will infect the cells there, and then those cells will start making dopamine. So I'm going to make a new set of dopamine producing cells. They're not nerve cells, and they're not the type that are lost, but I'm going to replace that bit of the system by putting in a new gene therapy. An alternative approach, which has now just come back in, so this was 20 years ago, was to try and use gene therapy like deep brain stimulation. So in deep brain stimulation, as we're hearing, you put a little electrode in, typically into the subthalamic nucleus. You turn on the frequency, and it basically silences the subthalamic nucleus. And so one of the approaches has been, can I put a gene into the subthalamic nucleus that turns it from producing positive signals to becoming silent? So can I actually switch it off by changing the identity of the nerve cells there? And that has been done in a trial, and that has recently just completed a new trial last year, and we've yet to see the results on that, but it sounds quite encouraging. And then the real new kid on the block for this is the idea of using gene therapies not to actually replace dopamine directly or to rescue, regrow that system or to fiddle with the circuitry, but to get at what goes wrong in Parkinson's by replacing key things that underlie the problem with developing Parkinson's in some people, but might be true for all people. Now, I don't have time to go through all of these, so I'm just going to pick on a couple of these to illustrate the point. So I'm going to start with growth factors, because this is now back in the clinic. There's a major trial ongoing with this, and this is funded by Big Pharma. So the principle here, as I say, is to put in a fertilizer that will regrow a dopamine system that's dying off. And the factor which most people have got attracted to is this thing called GDNF, glial cell line-derived neurotrophic factor, that was discovered in 1993. It was discovered in a company, and that created some problems because the company actually owned this molecule, even though it's found naturally. And some of you may remember that there was quite a lot of problems around this in the first part of this century, where trials were being done with not the gene, but infusing the protein. So they directly infused GDNF into the brain in patients. Some improved, but when it came to a more formal trial, it didn't improve. The company took the growth factor off the market. People then protested to say, well, I benefited from it, can't I have that growth factor? The company said, well, I don't care, it's ours, we've invested in it, we're canning that program. So that was the end of it. So this caused quite a lot of controversy, and I'm going to come back to this because ownership of some of these therapies and how they're actually brought into the clinic has created some major problems, and we've been caught out by a company that produced a gene therapy that then went bankrupt. But nevertheless, for our story today, GDNF is the growth factor we're interested in. And then about 15 years ago, gene therapy trials were started with this growth factor. Now, unfortunately, they couldn't do gene therapies with GDNF because GDNF was owned by the company. So they had to make a gene therapy that something was similar to it. So they made it with this thing called nurturing. And what they showed in this trial was when it's quite ethically controversial, some of this. So what happens in these trials is that, I don't know if the pointer's working on this, doesn't seem to be, is essentially what happens is people get randomized to have surgery or not surgery. So if we were doing this trial here, this would be a big trial, but essentially everybody goes to theatre, everyone has a hole drilled in their head, half of you get a needle put in and a gene therapy injected, and half of you, they just pretend to do that. So a lot of them have scripts, they have the noises of the devices, so you don't know whether you've had the treatment or not. The only person who knows is the surgeon. So in this trial here, in that top figure, there is one group that had sham surgery, so they just had a hole in their skull, so if they felt it, they thought there's a hole there, so someone's been there. But only half of them had the growth factor in, and at 12 months, the red and the green lines are identical, so they were no different. So it made no difference putting this growth factor in. Everybody seemed to get better. But when they then followed people a bit later, out to 18 months on that bottom graph, you could see that the group who'd had the active treatment were a bit better. So this implied that it might work if we waited a bit longer. So what the company then did, which was Serogene, was they did a new trial where they delivered it at a higher dose. They delivered at 2 sites to the brain because they thought that might be critical, and they waited longer, and they found it made no difference. So the conclusion from these trials was putting in a growth factor into the brain was not helpful in people with Parkinson's. Now, there may be all sorts of reasons for that. One may be it just doesn't work. The more pressing point, I would say, is that you're trying to rescue a dopamine system that has already disappeared. So the analogy I have is I can have the best fertilizer in the world, and if I put it on my flower bed to treat my roses, but there's no roses in the flower bed, nothing will grow. And there's nothing wrong with my fertilizer, there's just nothing to grow. So the question is, are you trying to rescue a system that now has essentially disappeared, so nothing can grow back? Had you given the right dose, and had you given it to the right type of patient? So this caused basically the field to grind to a halt about 10 years ago, that this therapy was of no use. We then set up a workshop to try and identify all the factors which we knew or didn't know about this. And on the back of that, a new trial has begun by a company called AskBio, who are part of Bayer, which is a big pharma company. And they had their preliminary results that they released last year, showing when they put in a gene therapy to people with Parkinson's, it actually significantly improved their condition in this trial. So this GDNF gene therapy trial is now coming back to clinic in a big formal trial. I don't know whether it's coming to Australia, but certainly in the UK, this has now come back to clinic. So growth factors for Parkinson's are still ongoing. Now, I'm not going to talk about the dopamine therapies where we give people genes to infect cells to produce dopamine. All I will tell you is that the last trial we did of this, which was with a gene therapy with dopamine, we treated 6 patients, we saw some effect, and then the company went bankrupt. And this created major problems for us because we couldn't get the data from our patients because the data was on a database which was in an office in America, but there's no one in the office because the company's disappeared. So now we have a problem because we've done a trial. We have patients in a trial. They've had an irreversible therapy put into their brain. The company that gave it has disappeared. Who is responsible? We then tried to get the notes from where they'd been stored, the actual handwritten notes. The problem there was they hadn't paid the storage fees. So we couldn't get the notes, so then we paid the storage fees. We've got the notes, and now we're looking at it. But then the question arises, if something goes wrong in the future, who's responsible? So this is a bit of an issue in this field because the development of these gene and cell therapies is incredibly expensive. So it's not something that can easily be done in universities. It has to be done with companies. And if companies only have one product, they're, especially in the current climate, they are susceptible to running into problems. So I'm not going to talk about that, but I just wanted to mention another approach which is now coming to clinic and has been in the clinic probably for the last year or so, which is the idea about putting in a gene to replace something that's missing. So yesterday I was talking about this particular form of Parkinson's where people inherit one abnormal gene in this thing called GBA. About 10% of people with sporadic Parkinson's have this. And this gene, if you inherit it, reduces the level of an enzyme, and that enzyme is probably also reduced in people who don't have this mutation in Parkinson's. So this enzyme deficiency is associated in some way with getting Parkinson's, and it seems to not only increase your risk, but seems to also mean that the condition moves more quickly. So a company now, and several companies, have taken this as a way of trying to treat Parkinson's. So the argument here is I will inject a gene which will get into your brain, and what that gene will do is it will produce this enzyme, GCAs, which you're low in. So I will now ramp it up so it goes to normal levels. If that's involved with your Parkinson's, it should sort out what's going on and make you better. So this trial is ongoing. This is where they've injected it into the fluid around the brain. So this is called intracisternal. So they go at the bottom of the skull, and they've injected it, and they're following people over time. And there are now many companies that are doing this. And in fact, there's a company I'm meeting next week from California called Capsida, who now think they can do this by injecting something into the bloodstream, and the gene will selectively be taken up into the brain. And then it hopefully will restore the enzyme back to normal and slow down and treat people with Parkinson's. This same strategy is also being done with Parkin that we were hearing about earlier. That this, people who have the Parkin mutation have a lack of this protein called Parkin. So the approach here is to inject a gene to replace the missing Parkin, and people with Parkin tend to have a very selective disease around the dopamine system. So it makes sense to try and inject it into that site, and the company developing this think it also may be relevant for people with sporadic Parkinson's. So gene therapies have suddenly become quite popular in Parkinson's around growing the system as well as getting to the underlying pathology. So to conclude on this bit, we are now in a new phase of growth factors for Parkinson's. You can often read that they've had their day, but they are actually back, and I think they hold some promise. As I say, we're also now looking at gene therapies that are designed to actually not just rescue the dopamine system or make the dopamine system more efficient. They're actually trying to get to the heart of what goes wrong in Parkinson's to actually slow it down. And as some of you know, these gene therapies in other areas of medicine have been quite transformative. So things like sickle cell, haemophilia, some of these types of treatments now have completely transformed what we can do. And as I say, we're also revisiting this idea of changing the output of the subthalamic nucleus. One of the problems with this field is that people slightly rush ahead of where they are. So one of the things we do not know when we infuse these gene therapies into people's brains is how much. We've no idea what the dose is. You can do it in a monkey, but a monkey has a fifth the size of the brain of a human. So even if you do it in a non-human primate, we still don't know what dose that would translate to in a person. We don't know what stage of disease. We had a recent workshop where, again, we tried to take this forward so that we had a clearer view of how to do it. When we do it in the disease course, I think, as I say, is critically important, and earlier is probably better for growth factors. You want to grow things whilst you still have a system to grow. And the other problem, of course, is if you're trying to use a gene to deal with all the problems in Parkinson's, how do I get it everywhere I want? I'm going to inject it in the brain, but it's only going to go in that bit of brain. I need to get it distributed over the brain, which is why this new gene therapy, which could be given via the circulation and selectively taken up into the brain and therefore help all cells in the brain that are unwell, could be a revolutionary way forward. And as I've learned to my cost, going into these trials with small biotech companies can create all sorts of problems. And of course, you're dealing with patients' expectations, and they are naturally nervous as to why the company is gone. And people think there must be a safety issue and such like. When in point of fact, it was merely around finance. And the final question, which is always true of all of these therapies, is if these therapies work, because they're gene therapies, they're likely to be pretty expensive. If you can give them systemically, that's not a bad problem, but if you're having it injected in your brain, that's not such a non-invasive approach. So there will be questions about whether these can compete. So as I'll come on to talk about dopamine cell therapies, you could ask yourself, would I like an injection of a gene therapy to regrow a dopamine pathway that has already declared itself wanting to die because I've got Parkinson's? Or would I just be better off getting a bunch of new dopamine cells to replace those that are lost? So these are the type of questions we have to sort of face and the community have to face, because ultimately, are these going to be competitive and can we afford them? And as you're probably aware, some of the new, very advanced therapies for certain diseases are colossally expensive. So for certain forms of childhood motor neuron disease where they have gene therapies, the cost of that is about £3.5 million, it doesn't really matter what currency it is, about £3.5 million, dollars, euros for a single treatment. So that obviously is life-saving for the child. The question is, what are we prepared to pay for some of these therapies when we move them to Parkinson's? So now I'm going to change tack and just talk a little bit about dopamine cell therapies. So the principle here, and this is dopamine cell therapies. So there are lots of stem cell therapies out there that work in ways which are not around dopamine replacement. The strategy here is that you lose, as I say, 250,000 dopamine cells. So I'm going to give you back 250,000 healthy new dopamine cells of the type you lose in Parkinson's, which are this very selective group called the A9 nigral neurons. So why would I bother? We've been hearing over the last day and a half, we have tons of very good drugs and treatments for Parkinson's. So why would you even consider doing this? Well, there are certain advantages to it, which if this worked, would completely transform what we do. So the first thing it would do is, if it worked, would be replacing dopamine where it's lost in Parkinson's. So I'd put it the area where it's lost, which is in the putamen. So all of the problems that we see with drugs stimulating other dopamine systems, so the impulse control disorders, some of the neuropsychiatric problems, hallucinations, they would all disappear because you're not taking any dopamine which are stimulating intact dopamine systems. You're simply replacing the bit that's lost. So you would avoid all the off-target complications you see with the oral medication. If we put it back as a dopamine nerve cell, so it releases dopamine in the normal way, you won't need to take L-DOPA drugs, which don't work in a physiological way, so you won't get any dyskinesias. If you don't get any dyskinesias, you don't need deep brain stimulation and all the other pump therapies. And it has the potential to be a one-off treatment. So if this really worked, the future would be you turn up, you're diagnosed, you have a test response to dopamine drugs, you have a transplant, that's it. That bit of your Parkinson's has been sorted. That's not curing it, because the disease pathology carries on and it will affect other sites. But essentially, in theory, you won't need to take any drugs, and that is that part of your problem sorted out. So if that's the case, what could we use as our source of dopamine cells? Well, over the years, almost anything that produces dopamine has been tried, not in the lab, but in patients. So in the 1980s, you have a gland near the kidney called the adrenal, which produces really no dopamine. But anyway, people thought it produced some, so they took it out, shoved it in people's heads. Hundreds of people had that. But actually, what proved to be the most successful was a very controversial source of cells, which were the developing dopamine cells from the fetus. So in this case, what happens is a woman decides to terminate her pregnancy. That tissue is donated to medical research. They can't stipulate in which way. We then collect that tissue. We dissect out the bit from the dead fetus where the dopamine cells would normally develop into the nigral cells of the adult brain, and we then inject that into the brain of people with Parkinson's. So this pioneering work really took place in the 1990s, and I'm going to come on to a trial we've just completed. And when you think about it, you think, this is actually pretty bonkers. So I'm going to get some dead human fetal tissue. I'm going to ram it in the head of someone with a late onset degenerative disease, and what will be the outcome? Well, it would die. I mean, why would it survive? Well, in the preclinical models, it worked very well. So that encouraged them to go to clinical trials, and as I'll come on to point out, they did produce very variable results. But when it worked as a proof of principle, boy, did it work. So this is two patients from London who went to Sweden, and they had transplants of this human fetal midbrain tissue into their brains. So unfortunately, I can't point with this, but at the point at which they had the first transplant, they'd had Parkinson's for 10 years. So the scores going up, are the thing called the Unified Parkinson's Ratio Scale. The higher you are, the worse you get. So these people had moderately advanced Parkinson's 10 years into their condition. They had a transplant of fetal dopamine cells on one side. They then had the other side transplanted with fetal dopamine cells. A few years later, clearly their motor scores had come down to the level when they first presented to their neurologist or specialist 15 years previously. And then, as you can see, they just remain as a flat line for 15 years. So at the end of this graph, both of those patients have had Parkinson's for 35 years. They have normal dopamine levels on their brain scans, and they're on no dopamine medication. So, I mean, that's quite impressive, really. Now, I have presented this at other meetings, not surprisingly, and some statisticians said this could be a placebo effect. I said I didn't really care. I mean, if that is the size of placebo effect I can get, then bring it on. I don't really mind. And so you can get long-lasting clinical benefit, and the scan gets back to normal, but does that actually mean the cells have survived? So this was a patient, another patient, who also had a similar course, but eventually then went on to get Parkinson's dementia in their 80s. But they had a transplant on one side of the brain. So in the B up there, if you can see it, so you've got the sort of schema of the brain. The one next door, which has nothing on it, is the side of the brain that received no transplant. The brown is the dopamine. So in that side, they had no transplant, nothing happened. Everything else you can see are dopamine cells that have been put in a quarter of a century earlier. So even though they're being put into the environment where people have Parkinson's, they can survive long term. So what these studies have shown is that you can get long-lasting clinical benefits with restoration of dopamine back to normal, with evidence at post-mortem that those cells have survived and innervated the brain normally. Now, subsequently, this went to America, where there were some trials that were undertaken which were perhaps not as thorough as they could have been. And the consequence of that was that they found that their transplants did not work, and that patients developed some side effects, including these dyskinesias, which you obviously see in people who take L-DOPA, but now you were seeing them with the transplants and no L-DOPA. So this created something of a problem. So by 2003, when these second trial had published, the perception in the world was that cell-based therapies were a waste of time. They didn't work because the trials in America showed it didn't work. That's fair enough, but you have to explain that. So, I mean, why have those people done so well? So we then tried to analyze all the data that we could to find out why some people did better than others, coming a bit to what Laurie was saying this morning about can you use this sort of data to work out who does better. And what we found was if you look at all the trials around the world that had done this, a third of patients had done extremely well. Now, obviously, that's not good enough. I can't take you to theatre and say, look, you've got a 1 in 3 chance of actually benefiting from this. Otherwise, you've got 2 or 3 chance of it not working. That's not competitive. But what did it enable us to do is it enabled us to work out who might be best suited for this, coming back to a point I was making yesterday about not everybody's suitable for every treatment. So in this, we thought younger patients who are less advanced, we thought we needed to probably put in more tissue. We needed to immunosuppress the patient afterwards, which some of the trials had not done, even though the tissue was not their own, and longer follow-up. So this formed the basis of a new trial called the Trans-Euro trial that was funded by the European Union. And so this trial was supposed to be, we grafted 20 patients, got the results, and then went on and did another 60 patients. That was the original plan in 2010. And this paper will come out soon, and it's taken me 15 years to do this trial, and I only ever grafted 10.5 patients. And the reason will become clear that we just simply could not get the tissue in order to make this a viable option. So what did we find? Well, what we found was it didn't really work. But it didn't work in the way that we thought, but we definitely saw something. So what we found was, with these patients, was that we'd followed them for a long time before we did the intervention. So these people were coming up to clinic every 6 months, and for 4 years, we just did standard measures. They then had a transplant. We followed them for another 3 years. So they're 7 years into this study. And the blue line, which is the control, are people who've had nothing. So we haven't done sham surgery. We just collected information from them every time they come up. And what you can see is they just get progressively worse, as you would expect. But the transplant group have a sort of point of inflection. So they sort of get a little bit better or they don't get worse. And that clinical response was also matched by a couple of other measures. One is if you had a transplant, you needed less medication, and the amount of time when your medication was not working, or the off time, had decreased. So what this implies is we've underdosed. We sort of got a bit of a response, a little less dopamine, and a bit less time off. And we also found this on the scanning. So again, unfortunately, I haven't got a pointer here that works, but the black line are people who didn't have a transplant, and their dopamine levels in their brain get worse. The red are the transplant line, and they've got a bit better. The dotted line at the top is normal. So even though they haven't got worse and got a bit better, we're still a long way from normal. But those with good eyesight can see that there's a couple of dots of somebody who's actually gone up into the normal range, and that person's motor response was very dramatic. They almost went back to normal. So what this implied was that we were simply not putting in enough cells to treat the patients. And we elected that we would need the tissue from 3 aborted fetuses to do this. We would like more. If we had decided to use 4 instead of 3, we would have done no transplants. And in point of fact, for the 21 transplants we did, 10 bilateral, both sides, 1 on one side, we had another 87 surgeries that were cancelled because we didn't have enough tissue. So if you were in this trial, you had a 1 in 5 chance of going to theatre on the day that we said you were going to go to theatre because we had a tissue. So this is just simply not viable as a way forward, and the results would also show that this is the case. So if you're going to get around this, you have to have a better source of cells, and this is where we use stem cells. So this has been transformative in the last 15 years. So in 2011-12, it was worked out how you could take a human stem cell, either from a spare embryo, embryonic, or from a patient themselves, so-called induced pluripotent, as I think Cedric was talking about yesterday, and you could turn it into dopamine cells. And over the last decade, we've shown that you can do that consistently. They survive, they don't form tumours, and they actually have functional benefit in animal models. So these have now moved to the clinic. And this is the current landscape of cell-based therapies. So in California, Aspen Neuroscience, I think, have done 7 patients. These are people who've had their own cells, so this is autologous. So in this case, someone would come, you take the skin, I grow the iPS, I turn them into dopamine cells, I pop them into your brain. So that's happened there. At Mass General, they had a single patient they reported a few years ago. They've now got permission to do another 7. Arizona State University, they're hoping to think about doing this in Parkin. They've got an iPS cell. Blue Rock and Bayer, I'll present their results in a minute because they're about to go to a phase 3 study, which I believe is going to come to Australia, that element, one part of it. We have our own trial in Europe, which we do between myself in Cambridge and my colleagues in Lund, and we work with Novo Nordisk. There's a group in Japan who are just about to publish their trial probably in the next few weeks in Nature, and then there's a group in South Korea who've also completed a trial. So this has suddenly become quite big business. And these companies are seriously wealthy. So the advantage of that is they are in this to make a difference to people with Parkinson's. So Novo Nordisk, who we work with, are the richest company in Europe. I mean, they're not just the richest pharma company, they are the richest company in Europe. And that's not because they work with us on cell therapies. They make Ozempic and the GLP-1 agonist, which everybody takes for weight loss. And so for our trial, we had this particular design. We grafted our first patients in the early part of 23. This was all staged so we could check it was safe. We did 2 more in September. We did 2 more in the Easter of last year, and the last 2 patients in September and October, and we will have our results later this year to see whether it is safe and we have any signal of efficacy. Blue Rock, who are part of Bayer, they have now completed a 12-patient trial. They have yet to publish the paper, but their 24-month data is quite encouraging. So they used 2 doses of cells. In those that had the bigger dose, which is dose 2, this Unified Parkinson's Rating Scale off score improved by 22 points, which is a fairly significant improvement. They got 2 hours better time on with their medication, 2 hours less off, and the part 2, which is more subjective, also improved. So my understanding is that they will now move to a 102-patient, double-blind, placebo-controlled trial to see whether this works, and if it works, they want that to be their marketing trial, and this will become a marketable therapy for Parkinson's. And they also showed some restoration on imaging of dopamine. So just to conclude with cells, there's a very clear rationale for why we use dopamine cell therapies. Dopamine drugs very well, and we've got very good proof of principle from using fetal dopamine cells. We tried to do that with a formal trial, and we didn't really get the results we expected, but fundamentally there are major logistical problems, which is why the world has now moved to stem cell-based therapies. But there are challenges with this. So the challenges are that we don't put it where the dopamine cells are lost. That may not be important, but is that a critical question? We cannot monitor these cells. So if I put in a new liver in you and you start to feel a bit unwell, I can look at your liver function tests, I can do a biopsy, I know the transplant's not happy, I could change the therapy. With this, we have no way of knowing. Once it's in, it takes a year, 3 years to mature, but we don't know if it's going to be rejected. And so that is a big problem. We can't modulate it once it's in, so it'd be nice to switch it on and off in terms of the dopamine. And also people have to take immunotherapy for a year after this, which in itself is not too bad, but nevertheless is a risk. Although if you believe, as I was saying yesterday, that inflammation is a big problem in Parkinson's, then being on immunosuppression for your transplant might actually be a good thing in its own right. So with that, I'm going to conclude and say that there is a great deal of interest in rebuilding the dopamine system with GDNF. There's no real interest in using gene therapy to make dopamine in cells, but there is a lot of interest in using cells to do that. The gene therapy around EBS, as I said, is just finished. There's lots of work around dying cells. And the sort of final point I want to make, which really sort of picks up on various conclusions we've had at this meeting, is that actually we talk about these as all being mutually exclusive. And ultimately, I would say, in the future, you turn up with your Parkinson's, assuming we haven't been able to prevent it. I shove a bunch of new dopamine cells in your head. I'll give you a cocktail of drugs to slow it down. Thank you very much. I'll go and sort out another disease. And just to tell you how much this involves, so this is the TransEuro team. This is the STEM PD team. So all of that down there is the Novo team that helps us, which is about 2 to 1 in more than we have. So every time I have meetings with us, it's always the same old us, and they always have these huge teams, and my lab, and with that, I'll finish. Thank you.